Hello, we're glad you've joined us for this live webinar, Ultra-Sensitive Single Molecule Array, Technology to Revolutionize CNS Biomarkers, presented by Dr. Andreas Jeromin, PhD, Chief Medical Officer of Quanterix Corp. I am Marjorie Torres of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Labroots. To learn more, visit www.labroots.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I now present today's speaker, Dr. Andreas Jeromin. Dr. Andreas Jeromin has more than 25 years of experience in CNS clinical diagnostic and companion diagnostics and co-authors more than 150 publications. He is the founder of Atlantic Biomarkers, Inc. and currently the Consulting Chief Medical Officer to Quanterix Corp. And members of several biomarkers, CNS disorder consortia, and working groups. I will now turn the presentation over to Dr. Andreas Jeromin.
Thank you very much. It's a pleasure to talk to you today, and welcome. So the title of my presentation is Single Molecule Array Technology to Revolutionize CNS Biomarkers. And as you've heard, I'm the scientific and medical advisor to Quanterix Corporation based in Lexington, Massachusetts. So what I've done in this slide deck is basically give you an overview on the technology, which is the single molecule array technology, combined with a few case application in the CNS biomarker space, in particular with regard to blood-based biomarkers. So what you see on slide number 2 is now a snapshot of our customer base. The Simoa technology has begun to be commercialized in 2012. Quanterix is in existence since 2007 and just celebrated its 10-year anniversary as well. And this technology really has been very rapidly adopted, as you see on this slide as well. So we have a number of key academic partners, research institute listed on the left part of the slide, a number of biopharma companies, names you're maybe very familiar with, and a number of contract research organizations and other institutions who have utilized and continue to utilize this Simoa technology. So as of today, there are about 150 instruments, and you'll see what I mean by instruments in the space, and we have a very rapidly developing menu of biomarker assays, in particular in the central nervous system space. So on this particular slide, what is kind of the opportunity in our perspective for the Simoa technology? And I should say that in the first application, it's an ultra-sensitive detection technology, in particular for proteins, but it can also be used for the detection of nucleic acids and other forms of molecules as well, if one has a capture reagent and the detection reagent available as well. So in our view and the view of the field, we can compare this to what we call the iceberg analogy here. There's really just a small number of either approved or cleared tests from a regulatory perspective as in diagnostic use, but there's also a very small number of peptides or proteins which are detectable within the entire proteome of about 220,000 independent open reading frames as well. And one of the limitations, or maybe the limitations in the space, is the lack of what we call sensitivity to detect these, in particular, low abundance molecules as we find them in blood as they relate, for example, to central nervous system disorders. And this is shown here in the iceberg analogy, that we really just see the very tip of the iceberg with current technologies, and there's a great need to improve in the sensitivity of our current immunoassays to push the detection limit down to what we call the single molecule level as well. On the next slide is a representation of really the seminal publication from 2011 published in Nature Technology, which basically depicts on the left-hand side in the cartoon the principles underlying the single molecule array, or what I call in short Simoa technology as well. On the upfront side, so the way we detect a given analyte down to the single molecule, this is a very traditional sandwich ELISA. So we're capturing our analyte of interest with a capture antibody, which, as you can see in the slide here on the left, is coupled to a bead. And then we're detecting that analyte with the detection antibody, and fluorescence is our quantification signal here as well. Unlike a very traditional plate-based ELISA, so those people who are familiar with the 96 well format of an ELISA, we don't detect these molecules within the bulk solution of a microwell plate, but we now isolate these beads who have captured down to a single molecules into a single well, and that's kind of depicted in the cartoon here to the left, where you see this well. This well is designed from a size perspective that it can just hold a single bead with, in theory, a single analyte as well. And by isolating now these captured analytes on a bead into a single well, we dramatically increase the signal to noise ratio and the detection capabilities and sensitivity of a given ELISA. On the next slide, you see this again depicted in a different way. So you have your incoming matrix, as we call it, to be analyzed. That could be blood, could be whole blood, could be EDTA plasma or plasma, could be serum as well. This is, we take a sample out of this tube, typically 25 microliters, incubate this with our capture antibody coupled to a bead. This is what you see here down to the right. And then basically come in with the detection antibody, and then we isolate now these sandwiches who have detected a single analyte now into these single wells, which are actually arranged on a disk, which is typical Blu-ray technology, which was adapted now to build these discs as well. And these have 24 different locations on the disk. And there the beads are pushed now into these wells as well. So you have empty positions here, the majority of empty. If you have low concentration, then you have single wells which are filled with the single bead. And if you see to the far down right, now these single wells which are captured or are filled by a single bead are lighting up if they actually contain an analyte of interest or multiple analytes of interest. So down at the very low concentration end now, we are typically in what we call the digital mode as well. So we typically actually count the number of beads who either have a signal, we call them on signals, or those which we don't, and we actually form a ratio, and that's our quantitative readout signal. We call it average enzyme per bead. And here is a comparison to the typical traditional 96 well ELISA format, which is drawn on the top. So this is what we call the analog mode, because as I said, you detect here molecules in bulk. Typical reaction volume is your 100 microliters, so 100 times 10 to the minus 6 liters as well, and you are limited really by diffusion in this 100 microliter volume within a given well. This is in contrast to the Simoa technology, which is, as I said, in the digital mode at the low concentration range of molecules as well. So here now you have a reaction volume of 50 times to the 10 to the minus 15, because that's the volume of a single well, which will hold a single bead. It's 50 femtoliters. So now, in comparison to the 10 to the minus 6 times 10 to the minus 15, you have a 10 billion times smaller reaction volume and increase in sensitivity or signal over noise as well. So for this technology, one molecule is needed to reach the detection limit here as well. Obviously, if you have more and more molecules now being bound to these beads, and beads fill these micro arrays here as well, these arrays of single wells, you now transition into the analog mode. So you have ultra sensitivity in the digital mode. The more molecules you bind, you transition into the analog mode as well, and this gives you a dynamic range of about 4 to 5 logs, as you see in a traditional ELISA. This technology can also be multiplexed as well, which is depicted on this particular slide. And the way we multiplex the Simoa technology is basically encoding another color onto these beads. So now, in addition to your fluorescence signal you take as a quantitative readout, you have another color, let's say yellow, red, or blue, or green, as you see here as well. And depending on the color of the bead, this now encodes the analyte you like to capture. So multiplexing means you can detect and quantify multiple analytes within a given sample, i.e. it's multiplexing. And by basically encoding another color on the beads, now you have another color which is encoded or encodes your analyte of interest to be detected, which is determined by the capture and the specificity of the capture and detection antibody coded onto these beads. So you not only have ultra sensitivity for the Simoa technology, you also have capabilities of multiplexing as well, and you have the capability of actually developing your own assay as well, if you have access or have already developed a sandwich ELISA, because this is what we call an open platform. So Quanterix does provide a solution for custom assay development, either done in-house by Quanterix or done by our customers who have adopted the technology as well. And we call this custom assay development solution homebrew. So you as a customer or potential customer are provided with the beads to conjugate your capture antibody or reagent, and then with the chemistry to biotinylate your detection antibody to be used in the Simoa assay format as well. On this particular table, you see a comparison to some of the traditionally known ELISAs or developed ELISAs, commercial and diagnostic grade in nature. I just want to point out a few. So you see here tau, and we'll talk about the microtubule associated protein tau in a few minutes as well. We have typically sensitivity in what we call the femtogram per milliliter range, and that compares to, for example, a currently research use only and diagnostic assay developed by Inotest or for G-Review in a 3,500-fold increase in sensitivity as well. Another example is an assay for the HIV capsid protein 324. The Simoa LOD you see down here is 0.0025 picograms per milliliter. Conventional clinical diagnostic assay developed by Ortho Diagnostic is 15 picogram per milliliter. And I should say that this particular assay developed on Simoa for P24 actually utilizes the same antibodies employed in the Ortho assay. So it's a direct comparison as well. As you see on this particular slide, there is a 6,000-fold increase of sensitivity here as well, if you look at this particular number as well. And this actually makes this assay now the 224 assay, and this is a detection of protein. It's a capsid protein, as I said, of HIV, similar in sensitivity to nucleic acid technology such as PCR. The way the technology is currently being offered is as an automated standalone instrument as well, with the idea to allow for the instrumentation, which is the size of a fridge. It's a fully automated instrument. You provide your samples in tubes or a microwell plate. Then the instrument runs either the assay is provided and is commercialized by Quanterix or your own in-house developed assay, as I said, through the homebrew program or the homebrew assay. And then 45 minutes and plus thereabouts, the first results are provided, and you basically obtain results in form of a spreadsheet. So it's a very traditional clinical chemistry analyzer type of arrangement. What you also see on this slide is, i.e. the kit component we'll talk about in a second, and then the disk I refer to, and these disks contain now these 24 positions of these arrays of the microwells we discussed a few slides ago. So the typical workflow here on the left is in step 1 that you load your consumables, either cuvettes, the kits, and then the reaction is performed in cuvettes. You load the disk, as I said. You select your assay. Either this is an assay you purchase from Quanterix, you develop your own, you load your samples, and then in 45 minutes plus, so typically 75 minutes, your first results are obtained. And step 5 is basically data transfer and data analysis. From a CNS perspective, we do have a number of key partnerships in place to really provide qualified reagents as well. This includes a company in Belgium called ADX Neurosciences as well. We're working with a company in Virginia with biomarkers. We have a supply agreement in place for PBI-related biomarkers as well, and then with non-diagnostic we've licensed for neurofilament light chain, and neurofilament light chain is a marker I'll discuss. The neurology pipeline you see on this slide, just to give you a feel of the depth of menu, is really what we would call first in class. So we have various assays for key biomarkers. I don't have the time today to go through all these markers, but I'll present to you some data for total tau. We have now assays for neofibrillary activity proteins as well. Neurofilament light chain, I will show you some data we have developed and made available assays for mouse tau, Parkinson's disease related biomarkers such as alpha-synuclein as well, TDP43 and BDNF, which is important as a biomarker in psychiatric disorders as well. Importantly enough, coming back to this concept of multiplexing, we have begun now to commercialize markers for a combination of various of these markers. An assay to be released in April of 2017 is a marker, what we call traumatic brain injury marker, a 4-plex containing total tau, GFAP, UCHL1, that's a cell body injury marker as well, and neurofilament light chain as well. And as you see, we also have begun to and will make assays commercially available for various synaptic post or presynaptic proteins as well. In addition, we have a large menu for cytokine markers as well, and for various cardiovascular and oncology markers as well. But the focus of today's presentation is on CNS biomarkers. So in the remainder of this presentation, I will give you an overview of some of these applications in the neurology space. There's more than 50 publications now with regard to Simoa and CNS applications. We don't have the time, I don't have the time to discuss them in depth today, but at the end of the presentation, you'll see my contact information, so please feel free to reach out if there are examples that are not covered today, or you have additional questions with regard to the presented publication. So, and depicted on this slide is kind of what we think the flow of biomarkers is from the brain into what we call the cerebrospinal fluid surrounding the brain and the spinal cord as well into blood vessels. While it's not completely understood what some of these transfer mechanisms are, it's believed that the blood-brain barrier in preventing the majority of proteins to be really basically flowing from the brain's cerebrospinal fluid into the blood. There may be other clearance mechanism in the brain, such as the lymphatic system as well, which will help with the removal of, let's call it, toxic proteins or waste proteins from the brain into the periphery as well. And the idea is that because of this filter here, the concentration of some of these analytes are in blood are obviously much lower than in brain, but they are also much lower than in cerebrospinal fluid. And what the Simoa technology really has enabled over the last few years is now to move the frontier and advance the frontier of CNS biomarkers from CSF into blood, and to show that this has clinical useful applications as well, and some of the application I'll present to you in the coming slides. Just to kind of give a bit of a feel how much is there actually present in blood. And this is a slide provided by Glenn of University of Gothenburg to me. I wanted to draw this analogy. And so this analogy is now drawn for neurofilament light chain here. This is a component of axons as well. This in plasma is present at levels of about 10 picograms per milliliter as well. And in CSF, it's at about 50 higher concentration of 500 picograms per milliliter. So the 500 picogram per milliliter now here, if you convert this, this relates to about 500 milligrams per 1,000 cubic meter of a swimming pool, Olympic-sized swimming pool, the size of 20 by 25 and 2 meters deep as well. Obviously, in CSF and in blood, we have a majority of other proteins other than NFL, particularly albumin, which make up the majority of proteins, which we're not really so much interested in from a disease state perspective. So if we now convert here the concentration in plasma, the 50 milligram per milliliter, this makes about 50 tons per swimming pool. So to put this in a little bit more illustrative environment as well, for plasma and plasma NFL, the 10 picogram per milliliter here, they correspond to about 1/300th of a sugar cube of NFL per an Olympic-sized swimming pool of 20 by 25 by 2 meters deep. So in other words, it's a very small amount of sugar cube in this particular pool, and one obviously needs a very sensitive and precise technology such as the Simoa technology in order to be able to detect this very small amount of sugar cube or of analyte or biomarker of interest. So in the next slide, we'll discuss the use and the detection of a protein called tau protein as well, which is a protein which is mainly found in neurons as well. And it's a protein which is associated with microtubules and plays an important role in maintaining the cytoskeleton of neurons. Transport, which is really the key function of communication between neurons. The scientific evidence shows that after axonal injury, it depolymerizes and aggregates and is hyperphosphorylated as well. So over the last few years, there's really been a wealth of data generated to show how complex of a molecule tau really is. There's evidence of extracellular secretion of tau. It's not just found intracellularly as well, and this may be the toxic form of tau, which leads to spread of tau between neurons and glia cells as well. But it's also become clear that tau is heavily what we call post-translational modified, not just by phosphorylation, also by acetylation, glycosylations, and other forms of phosphorylation. It aggregates into oligomeric forms of tau, and it is also processed and cleaved by proteases at the N and at the C-terminus as well. So it's a complex molecule. Quanterix has developed an ultra-sensitive Simoa assay to total tau and also now phosphorylated tau, but these data here are for total tau as well. So what I show to the right, on the right here is basically an overview on the analytical characteristics of this assay. So you have a limit of detection of 17 femtograms per milliliter, and what we call a lower limit of quantification of 26 femtograms per milliliter as well. And for those participating or attending this presentation who are familiar as well, this meets all the criteria of a fit-for-purpose validation for a research use only assay. If we look at this in terms of precision, so if we run this now on multiple days and compare within a run on a day or between runs over a day, we see that typically these assays perform with a precision less than 10%. So in 2014 now, this is one example I will discuss today. A presentation was published, or data were published for the use of total tau in what we call concussion or mild forms of traumatic brain injury. This is now in Swedish professional ice hockey players as well. And these data were published in JAMA Neurology in 2014 by Henrik Zederberg and Leonard University of Gothenburg as well, utilizing the Simoa tau assay. what you see on this slide here on the left is basically a comparison now of plasma tau concentration in pre-season versus post-season concussion as well. And as if you compare the pre-season to post-season, you see that you have significant increases in the post-concussion period compared to the pre-season period. One of the unique features of this study is to actually do sequential and longitudinal draws of blood in concussed players and compare this to preseason. So on the left here you have preseason as well, and then you basically have a time course here now on the right over a period of 144 hours. So as early as 1 hour, 12 hours, and 144 hours. And what's plotted here, if you look at the mean values, a little bit hard to read. You actually see that as early as 1 hour in the concussed players, total tau was elevated compared to preseason, suggesting a potential diagnostic use of total tau in these professional ice hockey players as well. The question then arose, can this also be used to, can the total tau concentration or the kinetic profile of total tau also be used to inform what we call return to play? So when is it safe for a player to return to play without any potential long-term consequences of this traumatic brain injury? So on the next slide now you see a few case study out of this publication where basically we have plotted here again for different groups now. So those with mild forms of injury, more moderate forms of injury, and then more severe forms of injury based on clinical criteria, what the total tau profiles in blood look like. And in red here you have what they call type 3 in this publication. This is a severe form of concussion as well. And compared to the mild and moderate here, you have a more sustained and significant increase now in total tau over time before it returns to baseline as well. And this is also the player where, based on the severity of the TBI or concussion, it took the longest time to return to play. So there is the first suggestion of data here that total tau in blood, this is not measured in CSF, this is measured in blood, can be used when followed longitudinally as a potential measure to inform return to play. And this is something we have independently now confirmed in a very recent 2016 neurology publication in collegial athlete, not in professional ice hockey players as well. So summary for this particular slide is that plasma tau may serve as additional tool to enable return to play. Other markers, biomarkers of potential brain injury or potential biomarkers of brain injury are analyzed in this publication, such as a calcium binding protein known as S100 beta and neuron-specific analyze. And unlike tau, S100 beta and NSE did not seem to correlate to return to play time or decisions here as well. So switching now from total tau to another external protein as well. This is a protein known as neurofilament light chain as well. Neurofilament light chain is part of a family of neurofilament heavy chain. On the top here, neurofilament medium chain, and then neurofilament light chain vary by molecular weight, as you see in the slide as well. We have developed now an assay, an ultrasensitive assay for the detection of neurofilament light chain in blood, can also be used in CSF, utilizing a pair of antibodies which basically recognize the mid-domain of neurofilament light chain. These are antibodies provided by a non-blood. On this slide here now you see a comparison, and before I discuss the data, I wanted to make sure that you actually, so it's comparing what is plotted in the slide here is either a signal in the ELISA in comparison to the same antibodies employed in the Simoa. This assay actually was originally developed in what we call the home brew format. So it's a custom assay development, Quanterix has since commercialized this assay as well. And then both the ELISA signal and the Simoa assay signal are plotted against increasing concentration here of neurofilament light chain. And as you increase the concentration of neurofilament light chain as what we call your calibrator, obviously the signal in your assay would go up. And this is typically what we see a calibration curve here. So you see in this slide that for the ELISA assay, basically the curve ends here. This is where around here where the limit of detection is lying as well, where you basically have an increase, a 2.5 standard deviation increase over blank. In the green here is depicted actually where your clinical samples typically read. So with your plate-based ELISA, you're actually limited to CSF or higher concentrations of NFL, and you are unable to detect NFL in neurofilament light chain in blood. But the same antibodies were now deployed on the Simoa assay, which is shown here in the blue triangle, in the blue triangles here, and you see that you now extend the assay towards what we call the lower end of the curve as well, and now you're venturing out into being able to quantify neurofilament light chain in blood in the screen window here as well. So again, coming back to the theme of this presentation, you have now ventured and you have actually pushed the frontier from being a CSF assay only and being able to precisely quantify neurofilament light chain in blood. And I'll show you in the remainder of this presentation some of the clinical applications. What is the correlation between NFL in CSF and in blood? And this is one example of some of the clinical studies published here. It comes back to the discussion we had of the blood-brain barrier, of clearance of some of these proteins out of the brain and from them into neurodegenerative process otherwise. And yet you see in this slide plotting NFL versus NFL in blood versus NFL in CSF, you see that you have a very tight correlation between these two matrices, blood and CSF. You have an R square of about 0.97, which is obviously highly statistically significant as well. So good correlation between or excellent correlation, what you measure in CSF compared to what you measure in blood. What are some of the applications here now? And so this is a publication by Jonathan Rohrer now in frontal temporal lobe dementia as well. What's plotted here are controls versus various forms of frontal temporal lobe dementia, various behavioral variants as we call them, including a form of which has a motor neuron disease-like phenotype as well. And what you see here that all the concentration of neurofilament light chain is measured in serum now as well, compared to the controls are elevated as well. And you start seeing a differentiation of neurofilament light chain levels between these different variants of frontal temporal lobe dementia, in particular frontal temporal lobe dementia with a motor neuron disease phenotype. NFL in CSF originally was also used for the differential diagnosis of Parkinsonian disorder, so differentiate Parkinson's disease from what we call atypical Parkinson's disease, multiple system atrophy, MSA is one of these examples. And this is a clinically diagnostic relevant discussion. And what you see here is now neurofilament light chain levels now compared in controls in typical forms of Parkinson's disease, including PD here as well. But then the atypical forms of Parkinson, as I mentioned before, including MSA. And as you see here, all controls versus all other groups, the neurofilament light chain concentrations here are different. They're elevated compared to the controls. And also PSP, supranuclear palsy, MSA, and CPD is different from all other groups. So here now is again an example where previously in CSF, only detected neurofilament light chain can be used in blood, which is a way more accessible matrix compared to CSF, which is to be generated via lumbar puncture, can be used for the differential diagnosis of atypical Parkinson's disorders versus Parkinson's disorders, and this is will be implemented in the clinic in the not too distant future. A final example coming out of the Alzheimer's space. Here now as well, looking at plasma NFL and the correlation to various cognitive changes and imaging changes. This is a little bit of a busy slide, but if we just focus on the left here as well, this is a change of neurofilament light chain concentrations over time as a function of a measure of cognition known as the minimal mental state exam as well. And this now the neurofilament concentration in these different colors were grouped into different quartiles as well, to basically group Alzheimer's disease with the highest concentration of neurofilament light chain from controls and mild forms of cognitive impairment, what we call MCI. So in blue here would be the AD group as well with the highest quartile. And what you see here that there is a nice good correlation between the neurofilament light chain concentrations here as well in these various quartiles as a function of cognitive performance here as well. And this is also replicated on the right here with a different measure of cognition as well. So here we're measuring neurofilament light chain originally found in axons now being released upon neurodegeneration into CSF and blood. And we actually do show that this is a important indicator and correlates to changes and declines in cognitive performance, and also correlates, this is on the slide here as well, to various measures of neuroimaging as well. So we're measuring in the periphery, we're measuring low concentration of neurofilament light chain, and this is indicative of brain pathology and clinical changes in these participants, in these subjects as well. And obviously this has significant implications, not just in understanding these neurodegenerative diseases, but also with regard to the use of neurofilament as tool in drug development as well. So with this, I want to finish today's presentation. It was a bit of a snapshot of technology and clinical application. I think in summary, hopefully I've convinced you that Quanterix has developed a biomarker toolbox, giving you examples for the detection of various CNS-related proteins as well, for diagnostic and prognostic applications, and also in support of drug development. We didn't really talk much about the support of drug development, but I'm happy to have this conversation as part of the Q&A and chat as well. You'll see my email address at the bottom of the email. Please feel free to reach out for additional questions, publications, or any other remarks. And with this, I thank you for your attention.
Thank you, Dr. Jeromin, for your presentation. A quick reminder for our audience on how to submit questions: simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Andreas Jeromin will answer as many questions as time permits. The first question is, for which biomarkers do you see the greatest need in terms of sensitivity?
Thank you. So as I gave you a few examples here, the idea really is more, I think, conceptually that we're, as a field, we're coming to the realization that, like in oncology, these brain diseases are really molecular defined diseases. So we need, coming back to this toolbox idea, multiple biomarkers to really understand the pathophysiology, not just for diagnostic application, but also for the efficiency measurements of drug development, efficiency and safety measurements as well. So this idea of precision medicine as well to target specific drugs to specific patient populations, either in the Alzheimer's space, in the Parkinson's disease, traumatic brain injury, and other CNS disorders as well. Some of the biomarkers, I think I talked about this today. We didn't talk much about the peptides as well, where assays are available as well. We talked about total tau, neurofilament light chain, and I think a new category of markers, again, we didn't have the time to talk about today, are pre and postsynaptic markers, potentially as very early markers of disease processes as well. One of the keys, and I think this is another question of yours, is really to diagnose these diseases early when there is the highest probability of therapeutic success.
Other than blood, which are other fluids such as saliva you think are important for the detection of biomarkers, for which applications, diagnosis, and clinical trials?
So compared to CSF, the generation of blood, and there are various other, so compared to CSF, the collection of blood is less invasive, and there are technologies being developed to make it even less invasive than what we traditionally have known as the phlebotomy in the space. But I think venturing out into fluids which can be generated more in a home type of setting, such as saliva, is an important venue of research for the future. So is the collection of potentially tears, if we think about this from an ophthalmology perspective, looking in other matrices more in the periphery, I think, such as urine, is where really the Simoa technology is strategically positioned because of the low abundance of some of these biomarkers.
Looks like we have time for one more question. Which are the most important preclinical or clinical applications to you?
So again, one of the areas of application we didn't really talk about today is the use of the Simoa technology in preclinical, i.e. animal models, for example, where the cerebral spinal fluid is very limited from a volume perspective. So there are several applications now where the Simoa technology, because of its ultra-sensitivity, has been used to allow the detection of multiple biomarkers in CSF in animal models, for example, of neurodegeneration. The added benefit of ultra-sensitivity is the fact that we can actually dilute out the sample and still be able to quantify the analyte. And then we combine this with multiplexing. This is a win-win situation. From a clinical perspective, coming back to this idea of precision medicine and really diagnosing brain diseases earlier, to reduce some of what we call the diagnostic delay, but also to be able to really treat patients much earlier in the disease trajectory, I think is the future of precision medicine in CNS disorders.
I would like to once again thank Dr. Andreas Jeromin for his presentation. Do you have any final comments?
Hopefully this was informative to the audience as well. I look forward to future discussions, and as I said before, please feel free to reach out to me or Labroots for additional questions.
I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information he has provided at the time of registration. We would like to thank Labroots for making today's educational webcast possible. This webcast can be viewed on demand through July 13, 2017. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.